I'm going to present a work concerning genetic alteration that confers increased risk for development of melanoma. As everybody knows, melanoma, the major factor involving development of melanoma is UV radiation exposure. However, host factors, specifically genetic factors, also are involved in development of this disease. So better tools are needed to provide more accurate treatments and personalized patient management. And for this, the understanding of the molecular parts of the tumor that underlies melanoma oncogenesis may be important in order in several areas, namely diagnosis, prognosis, and treatment of disease. We worked in a molecule, a protein named cyclin D1 protein, and this protein is involved in the regulation of the cell cycle, of the cell proliferation and cell division. All cancer cells have unlimited replicative potential, so cell division is very important. The control of cell division is very important in the oncogenesis of all cancers, namely and specifically melanoma. So this protein, it's involved in the regulation of cell division and cell proliferation. We studied genetic variants in this gene, in cyclin D1 gene, and these genetic alterations that are very common in the population. This genetic alteration produces altered proteins that confer growth advantage for melanoma cancer cells. So individuals carrying these genetic alterations are at higher risk and have a higher probability of 80% actually to develop melanoma. These are our conclusions, our results. And so the understanding of these molecular variations, very common molecular and genetic alterations, can be very critical for early diagnosis, for better prognostic assessment and accurate prediction of treatment response. Furthermore, it can be once we define the patterns, the pattern of molecular alterations that are important in melanoma oncogenesis, we can identify and select high-risk individuals for chemoprevention trials. Thank you very much.